and not jerky and exactly what you wanted them to be. And again, the medulla oblongata does a lot of that vital function regulation there. Okay, so now we've looked at a response, we've looked at how it's integrated. Now let's look at some of these specialty receptors that help send the information in that needs to be integrated and may require a response. So we're going to look at both eyes and ears, hopefully as quickly as we can. Okay, it's just kind of a brief overview of some eye anatomy. Okay, first we've got the outer layer here, which is the sclera. This is the white part of your eye. Okay, all of this, it's literally, like I said, it's what you see and call the white of the eye. Okay, it's tough, it's strong, it helps the eye, it helps protect the eye essentially. Then you've got your iris here. That's the colored part of your eye. Okay, and that surrounds the pupil. The pupil is actually a hole. Okay, it is not a solid structure. It looks black because it's a hole that lets you see, lets the light get into the back of the eye. And so all that light gets absorbed, and so it looks black. So the iris is the colored part of your eye, and that'll change shape and regulate the size of the pupil. The cornea is the clear portion on the front of the eye here, okay, helping protect the eye in the front. And the cornea is continuous with the sclera. The cornea, like I said, is just clear because otherwise you wouldn't be able to see. And then you've got your lens here. Your lens will change shape, allowing you to focus the light. Okay, so the job of the lens is to focus the light as the light comes in, to bend those light rays and make them focus on the back of the eye. So it'll change shape to do that. The retina is where you're actually going to have the receptors and the neurons that are going to send that information then back to the brain. And then the optic nerve is the nerve that will take it back to the thalamus, and the thalamus will then decide it needs to go back to the occipital lobe. So let's look at some of these receptors that are going to help stimulate the eye. So we've got two basic types of receptors in the back of the eye. Okay, we've got the rods, which are more sensitive to the grays. Okay, and we've got the cones, which are ones that are sensitive to color. So like I said, we've got these two types of receptors in the back of the eye. The light actually travels into the front of the eye, has to go through, you see the light coming in here, has to go through all of these and stimulate way back here. Okay, so first it goes through all of here to stimulate those receptors, and then the message turns around and comes back this way to then head towards the optic nerve. Okay, so it does seem to work kind of a little bit backwards here. So like I said, we've got two kinds of receptors here, rods and cones. They are both what are called photoreceptors because they respond to light. Okay, so we've got two kinds of photoreceptors here. Okay, and they're both going to work in a very similar manner. So let's look at the rods. We're going to look at the rhodopsin and the rods and see how that helps turn this light into an image of what we actually see. So rhodopsin is a chemical that's found in the rods. So I've got my rod, sorry about that. I've got my rod receptor right here. Okay, and in these discs there, you're going to find this rhodopsin. The rhodopsin is made up of two parts, retinol and opsin. Okay, and so in response to light, the retinol that's in there will change shape. Okay, you can see here's one shape of the retinol. So this is the retinol shape. And here's the second shape. Okay, and so normally it's in this bent shape here, but when it responds to light, it will flatten out. Okay, and so when it flattens out there, it changes shape. That's going to generate an action potential. We're going to watch a short video that shows this a little bit more in depth. Okay, but that's what we're looking at here. Is we've got these pigments that change shape in response to light. The shape change will trigger an action potential. That action potential will head back to the brain. Okay, cones have a very similar pigment in them called photopsin that works in the very similar manner. Okay, so we're going to just focus on the rhodopsin here. Okay, but that's all we're looking at. And it will take, you'll notice here it takes enzymes to change the retinol back from the trans isomer into the cis isomer. And during that period of time where it's changing back is where you are unable to process another image. Okay, it's called bleaching. Okay, so when it changes the shape, it bleaches, quote unquote, bleaches the disc. That's why the real bright light very quickly bleaches everything and it makes it hard to see. Okay, it's kind of blinding. So let's watch, we've got a short video with eyes, and then we'll move on to ears.
Photosensitivity in animals depends on a family of pigments called rhodopsins. In the human eye, light must travel through several layers of neural cells before it can be absorbed by the rhodopsin in rods and cones. Rods and cones are also called photoreceptors. The rod cell, named for its shape, is a modified neuron that does not produce action potentials, but it does release neurotransmitter from its base, where it forms synapses with the next neurons in the visual pathway. Each rod has an outer segment packed with membranous discs. The disc membranes are stacked inside the rod cell outer membrane. In addition to rhodopsin molecules, the disc membranes carry G proteins, also called transducin, and molecules of the enzyme phosphodiesterase. The rod cell outer membrane contains a type of sodium channel that is held open by the binding of cyclic GMP molecules. The open sodium ion channels allow sodium ions to flow spontaneously from a higher sodium ion concentration outside the cell to a lower concentration inside the cell, thereby depolarizing the cell. Light sensitivity begins with the absorption of light by rhodopsin. Rhodopsin consists of a protein part called opsin and a light-absorbing part called 11-cis-retinal, which is cradled in the center of the opsin and bound covalently to it. When 11-cis-retinal absorbs a photon of light energy, the molecule rotates around an internal bond, straightening out, and becomes all trans-retinal. The shift in retinal forces a conformation change in the opsin that signals the detection of light. The photoexcited rhodopsin activates transducin by stimulating it to release GDP and pick up GTP. Activated transducin, in turn, activates phosphodiesterase. Activated phosphodiesterase then converts cyclic GMP to GMP. The reduction in cyclic GMP inside the cell allows the sodium channels to close. Pumps in the cell membrane continuously work to drive sodium ions out of the cell, so the cell can now become more negative. The membrane hyperpolarizes. Signal transduction cascades have enormous amplification abilities. In this example, each molecule of photoexcited rhodopsin can activate several hundred transducin molecules, thus activating a large number of phosphodiesterase molecules. The catalytic capacity of phosphodiesterase is great. One molecule can hydrolyze more than 4,000 molecules of cyclic GMP per second. Thus, a single photon of light can cause a huge number of sodium channels to close. To test how a rod cell responds to light, we can penetrate a single rod cell with an electrode and record its membrane potential in the dark and in the light. In the dark, the membrane potential is around -35 millivolts. This value is considered depolarized, that is, relatively close to zero. In the dark, when the cell is depolarized, it continually trickles out neurotransmitter from its synaptic terminals. A brief light stimulus triggers the membrane to get more negative, that is, to hyperpolarize. As the membrane hyperpolarizes, the cell releases less neurotransmitter onto the next neurons in the visual pathway. A brighter light stimulus results in an even stronger hyperpolarization and an even greater reduction in neurotransmitter release. Note that this is the opposite of what other types of sensory receptors do. They typically depolarize and release neurotransmitters in response to a stimulus. To be a system sensitive to light changes, the signal transduction cascade needs to turn off soon after the signal has stopped. With vertebrate eyes, the retinal and the opsin separate from each other soon after light absorption, causing the molecule to lose its photosensitivity. A series of enzymatic reactions is then required to return the all-trans retinal to the 11-cis isomer, which then recombines with opsin so that it once again becomes the photosensitive pigment rhodopsin. Additionally, the G protein has an automatic shutoff mechanism. After a short while, it cleaves its GTP to form GDP and thereby becomes inactive again. Without the activated G protein, the phosphodiesterase stops its activity. while another enzyme in the cell replenishes the supplies of cyclic GMP. The sodium channels open again, bringing the cell to its resting state.
Okay, so now that we've done the eye, let's take a look at the ear. Okay, so the ear is kind of broken into 3 clumps. We've got the outer ear, middle ear, and inner ear. The outer ear is what will contain what we think of as our ear, okay, with the pinna, the ear lobe, the auditory canal. The middle ear contains these little tiny bones here. So this is our eardrum right there, also called your tympanic membrane. All these little bones here are called the ossicles, the auditory ossicles. This eustachian tube right here, that's what connects our ear to our throat. That's part of why when we get sick, it can move all around our head. And then the inner ear and here, these are where the actual receptors are, both for hearing as well as for balance. Okay, so the hearing receptors are actually located in this thing here that looks like a snail. Okay, and your hearing receptors, they're a form of mechanoreceptor, which means they need a mechanical change for them to actually cause an action potential. The photoreceptors required a light input to cause an action potential. These need a mechanical or physical change to cause an action potential. For instance, these cells have cilia or hairs on the end, and the mechanical change is that these cilia or hairs need to bend. Okay? And so that's a physical mechanical change. Okay, and that was what will trigger the action potential. So these are going to work a little bit differently than the receptors in the eye. So as the sound wave travels into the ear, so the first thing we need is we need something to create the sound wave. So something is vibrated and creates the sound wave. And the outer ear's job is to essentially catch that sound wave. Okay, and it's going to catch that sound wave and direct it down the canal. Okay. So that's really the job. That's why your ears are shaped like they are, to help capture those sound waves and direct them down the ear canal to the eardrum. And the eardrum, or the tympanic membrane here, is going to seal that entire canal. So this entire canal is closed off. There's no leaks in there. And so when that, it's sealed tight just like a drum head would be. And so when that sound wave travels down the canal and hits the eardrum, it makes the eardrum start vibrating. And these auditory ossicles here, you can see how it touches the eardrum. And so these auditory ossicles start vibrating. It really concentrates these little tiny bones here. By really kind of narrowing down the sound wave, it really helps concentrate the sound and the vibration so that we can actually distinguish what it is. So these ossicles then transfer that vibration here to the inner ear. And the inner ear is actually, this is all bone. Okay, so it's like a snail shell bone. These guys are all bones, but those are important for balance. And so those bones, though, are filled with fluid. And so the vibration that's coming in here off of this, off of the stapes, off of that last auditory ossicle, is going to trigger the fluid in the cochlea. That's the snail shell. It's going to trigger the fluid in the cochlea to begin to vibrate. And so now you have this fluid that is making waves. Okay? And so if the fluid makes waves, and I've got these little hair cells, they're going to start to bend. And so now I get my mechanical or physical change, okay? And then that information is sent along the vestibulocochlear nerve back to the brain, back to the temporal lobe of the brain to be processed. Okay? So we've got one more video on that, and then after that video, we are all set with the nervous system.
The ear is responsible for translating variations in air pressure, whether from speech, music, or other sources, into the neural activity necessary for our perception and interpretation of sound. The ear can be divided into 3 principal sections: the outer ear, the middle ear, and the inner ear. Each of these parts performs a specific function in processing sound information. Sound waves are first collected by the outer ear, which consists of the external ear, or pinna, and a canal that leads to the eardrum. The configuration of the external ear amplifies sound, particularly at the frequency ranges of 2,000 to 5,000 hertz. Not coincidentally, most human speech sounds are distributed in the frequency range around 3,000 hertz. From the ear canal, the